So here is the short summary of apoptosis from the previous video. So it is a programmed cell death, which is a controlled process eliminating damaged or unnecessary cell. Damaged or unnecessary cell. So its function is maintains homeostasis, prevents cancer, and is important for development and immunity. Then it has apoptosis process has two pathways. The first one is your extrinsic pathway. Then the other one is your intrinsic pathway. Let us now look at the extrinsic pathway. Here, receptor is your death receptor. That is TNFR-FAS. Then trigger or signals is your external, is extrinsic signals, or the trigger is your external. So here steps is your binding of ligands like TNF or FASL to death receptors, TNF or FAS. Then second one is your formation of the DISC. That is death-inducing signaling complex. Then activation of caspase-8. That is initiator caspase. So before this, if necessary, flip protein will inhibit caspase-8 activation. Caspase-8 activates executional caspases directly or clips bid. Bid becomes bid. And this will link extrinsic and intrinsic pathway. Now, intrinsic pathway or mitochondrial pathway. Trigger is your internal signal like DNA damage or the oxidative stress. So steps is your activation of P53 in response to stress. Then release of back proteins which permeabilize the mitochondrial membrane. Release of cytochrome C into the cytosol. Then formation of apoptosome. That is APAF1 plus cytochrome C. Then activation of caspase-9. This is the initiator caspase. Then activation of executional caspases 3-7 leading to cell death. So in extrinsic pathway, this is your, it contains, so DISC includes death receptor plus adaptor molecule plus caspase plus ligand. So here, DISC complex consists of ligand plus its death receptor, adaptor proteins, and caspase 8. Then this death receptor, it's a homotrimer and belong to TNF receptor family, that is tumor necrosis factor receptor family. And this flip protein, they resemble caspase 8, but this flip protein, they lack cysteine in its active site. Therefore, they cannot cannot perform this protease activity and thus block for the apoptotic signal. And then regarding the intrinsic pathway, the triggers include hypoxia, high or extremely high concentration of cytosolic calcium, and viral infection, stress, DNA damage, or the developmental signal of the oxidative stress. And it includes like the redox molecule like NADH, NADPH, glutathione are oxidized and reactive oxygen species. Their concentration gets extremely high, and then which causes oxidation of lipids, proteins, or other molecules, leading to the disruption of mitochondrial inner membrane potential. And after that, this like the permeability transition pore is there, which is formed and consists of ANT. ANT is your adenine nucleotide translocator, nucleotide translocator, and VGAC, which is a voltage dependent anion channel. ANT is your transmembrane channel, which is for the export of ATP in exchange of ADP required for the process. VDAC is your pouring of outer mitochondrial membrane, and it forms non-selective pore through outer membrane. Then this intracellular proteins, they trigger apoptosis. These are the caspases or the proteases, and they have cysteine at their active site, and they cleave their target proteins at specific aspartic acid residue. Then these are the process for apoptotic inducing condition for the internal intrinsic pathway. Then we learned about extrinsic pathway. So the receptors include TNFR and FAS, then signal is from the external, and then binding of this ligand occur, formation of DISC, activation of caspase 8, and further activation of the executional caspase occurs, finally leading to the cell death from both of the pathway.